So what does this thing look like? So there are lots of DNA polymerases. For the most part, they all share this same basic architecture, which the crystallographers, who are very creative people, have called a hand. Okay? And if you see this, you think, wow, they're right. It looks just like a hand. Okay? But this isn't what a real structure looks like. This is actually one of the simpler DNA polymerase structures, and it looks like that. And now you see why they're very creative people. Okay? What they call the fingers is over here. Okay? The thumb here, the palm here. Okay? So, you know, it's sort of hand-like. You could say it's a claw. They say it's right-handed, not left-handed. There's an argument for why they say that, and that's because this, as I'll tell you in a moment, moves, and this stays still during the catalysis reaction. And so that's sort of why they call it the fingers, because our fingers move a lot more than our thumb does. And in that case, it's definitely right-handed. Okay? So where are the various parts of the DNA binding? Let's move this up. So the palm binds the primer template junction, and as part of that, the newly synthesized DNA. Okay, so over here, you have the newly synthesized DNA, the primer template junction. One interesting attribute of this structure, and the structure of all polymerases, is you can see this is the template immediately after the active site. Okay, so there's one base unmatched here, and then the rest of the single-stranded DNA has a 45-degree bend in the DNA after that. And that's important because that means the only nucleotide that's actually around the active site to be base paired with is the next nucleotide after the last one added. Okay? So, but the palm is interacting with this, and again, you know, there's all this other stuff that we're not going to go into detail, but the palm is considered this region right down here. Okay? So this sort of creates an interesting question, which is how does a protein bind all DNA? Because we want it to bind that sequence, whether it's GATC, whether it's AGAA, whether it's GGG, it needs to bind to that. So what part of the DNA would it be binding? Does anybody know? Yeah. The backbone. So one idea is the backbone, and it does. Okay? So it binds to a certain extent the phosphate backbone, which does not change regardless of sequence. But what if I told you its primary interactions are actually not with the backbone? What would it be binding then? You think it'd be what's in the major groove? What kind of information? If you want a sequence-specific DNA binding protein to recognize DNA, do you know what part of the DNA it binds? I'll give you a hint. It's what I just said. So it binds the major groove. Okay? And if we look at this illustration, you'll see why. Okay? The major groove has these very interesting patterns of hydrogen bond acceptors and donors. And they're very different. So here it's AADH, okay, and H is sort of a nonpolar hydrogen that can't really be involved. Here it's HDAA. Here it's ADA, and then it has a methyl group, which can have a very different kind of interaction. And then this one is the opposite, MADA. Okay, so depending on which base pair you make, you can read out all this information about which base pair it is. You can tell if it's a TA or an AT residue. You can tell if it's a CG or a GC residue by looking at the major groove. But that's not what we want, right? We like this minor groove, okay? And the reason is, well, the middle of the base of the nucleotides, sorry, the hydrogen bonds is different. The outside ones are the same in every base pair, and they're actually positioned in identical positions. So whether you have an AT, CG, GC, or TA base pair, you'll get the same pair of acceptors for hydrogen bonding in the minor groove. And in fact, that's what happens. There are interactions that are non-sequence specific, but Watson and Crick base pair specific in the minor groove. And we're going to come back to this because it turns out many proteins that recognize DNA non-specifically use these interactions to do so. Okay? And that is important because what that allows the polymerase to do is not only to recognize no matter what base pair is there, but it can also say, yes, it's a base pair. Okay? So if you have an AG here, these things are all screwed up. They're not in the right place, and it can detect that. Okay? Or if you have a CC or a CT or any number of other mismatched base pairs, that distorts where those two acceptors are, and it can say, that new DNA you just made, that's not right. Okay? And it can do things in response to that. So it's detecting not only all different sequences, but it can detect whether there is a base pair that's correct or a non-correct, non-Watson and Crick pair of nucleotides. Everybody follow that? Because we'll come back to that several times later on.